Okay, hi class. So we're going to be starting chapter 6. We'll be covering infection. We'll be describing some of the basic characteristics of bacteria, viruses, chlamydia, rickettsia, mycoplasmas, fungi, prions, and helminths. And then we'll describe some of the modes of transmission of the microbes. We'll describe factors that can determine host resistance and factors that contribute to the pathogenicity and the virulence. We'll discuss some of the methods that can prevent and control infections. And then we'll talk and describe some of the diagnostic tests for infections and some of the mechanisms that are commonly used to treat. We'll discuss antibiotics and antimicrobial drugs. So, what are microorganisms? Right? If we look at the word, we see micro there, micro like microscope. It means small living forms. And these include bacteria, fungi, protozoa, and viruses. Many of these microorganisms can grow in artificial culture medium. Some microorganisms are considered to be pathogenic and some are non-pathogenic. So the non-pathogenic usually don't cause disease, right? They usually don't cause disease unless the condition changes. Non-pathogenic would include the natural microbiome or the normal flora of the intestines, right? There's lots of microorganisms in there that work in harmony with the human body. They're often beneficial. Many of those microbiome or the normal flora produces B vitamins. They help with enzymatic degradation of a lot of the foods in the intestines to break them down, part of the digestive process. Then we have pathogens. These are disease-causing microbes. These are just some different types of microorganisms. You're going to hear me talk about some of these in the upcoming lecture today. You could see on the top left there coccus, which are usually round spheres. You can see bacillus. These are rod-shaped. If we look at the spirilla, these look like they're swirly or spiral-like. If you look at staph or staphlo, these look like they're clusters. If you look to the right of that at the strepto, these look like chains, right? So we'll go through some of these in just a little bit. Continuing with the type of microorganisms, I'm not going into too much detail here, just to look at the difference between the structure. And because the structure is different, they're going to function different. If you look on the left, these are some of the parts that you'll hear us talk about when we talk about viruses. You can see that there's a core protein in the center. You can see that there is a capsid or a nucleic acid capsid around that. We have an envelope, and then we have a glycoprotein. Here it looks like they're spiked. And then on the right is a fungus off to the right. And you can see that there are some spores here, chlamydospore, blastospore. So we'll be talking about spores as well. Bacteria are classified as prokaryotes. You'll hear of prokaryocytes and eukaryocytes. So bacteria are classified as prokaryotes or prokaryocytes. There's no nuclear membrane. There is no nucleus. And they function metabolically and they reproduce, but they reproduce and divide by something called binary fission. We'll see that on the next slide. They have a complex cell wall structure, but what's interesting is they do not require living tissue to survive. They can vary in size and shape. Some bacteria can form spores that I mentioned before. We'll talk about tetanus on slide 8 that's coming up in a little bit, but let's just look at the binary fission. You can see what's happening here. You have this vegetative form here. You can see the DNA is duplicating. Then you could see it kind of pinches off here and the cell divides, and then you could see they have these two identical daughter cells, and then each of those daughter cells continues to divide and multiply as well. That's called binary fission. You can see here, this is spore formation, and this can happen in some gram-positive bacteria, such as tetani, or tetanus, right? Clostridium tetani. So we have this vegetative form. You can see that the DNA here is starting to duplicate, and the cell membrane here starts to fold inward to seal it off, and you can see that it happens right there. There ends up being these multiple layers, these resistant layers that form this very strong coat around the DNA. And then eventually the cell membrane here starts to lyse and it just starts to break down, and the spore is released. And under adverse conditions, the spore remains viable in the dormant stage. And when there is a favorable environment within the body, the spore sheds the resistant coat and it can multiply and continue to divide. It returns back to its vegetative state and goes through the binary fission process all over again and repeats this step over and over and over again. Type of bacteria. Before we looked at some of the shapes, bacilli are the rod-shaped organisms. The spirals are the spirochetes and spirilla. These contain that coiled, spiral-looking, or even wavy lines. The cocci are spherical forms, or they're round. When you hear diplococci, those are spheres and pairs, or they can be in chains, streptococci, or they can be in clusters, staphylococci. They have a rigid cell wall. It protects and provides and gives us a very specific structure or shape to it. And the two types that differ in chemical composition are referred to as gram-positive and gram-negative. And whether it's gram-positive or gram-negative is very useful in giving us the selective antimicrobial therapy, because the treatment and therapy for a gram-positive will be different than what could be used to treat gram-negative effectively. The cell membrane is located inside the bacterial cell wall, and it's typically selectively permeable. The external capsule has a slime layer, which offers some additional protection. Sometimes they will have flagellae, which, if you think of anatomy and physiology, you learned of a flagellae that was attached to the body of the sperm to help it become motile and move around, so it provides motility for some species. And some have fimbriae, or pili. These are tiny hair-like structures that are found in some bacteria, typically found in the gram-negative. And they assist in the attachment to tissues, and they transfer the DNA to another bacterium. And this results in greater genetic variation. And this is the reason why we have different strains of bacteria. The cytoplasm contains chromosomes, right? Chromosomes is basically one long strand of DNA. There's ribosomes, there's RNA, and something called plasmids. These are DNA fragments that are important in the exchange of DNA to other bacteria. Commonly contains genetic information conveying drug resistance. It creates different strains, right? So these plasmids are just small fragments of DNA that gets exchanged. Now, some bacteria produce toxins, and there are exotoxins and endotoxins. Now, exotoxins, just think of that X, and if you turn it, it looks like a plus. So it's a gram-positive, right? It's produced by gram-positive bacteria. And if you look at the endo, the N in endo for negative, right? Good way of remembering that. So exotoxins are produced by gram-positive bacteria, and endotoxins are produced by the cell wall of gram-negative bacteria. The exotoxins, some of them are neurotoxins, which will affect the brain and the neurology of the individual, and some are enterotoxins, entero from the gastrointestinal tract. When someone has enteritis, it's usually inflammation of the intestines. The endotoxins, which is released from the gram-negative bacteria, will result in fever, weakness, can often create these cardiovascular effects. And the vasoactive components, vaso, right, blood vessel, the vasoactive components can actually create endotoxic shock. Enzymes are important because the enzymes can damage the tissues and actually promote the spread of the infection even further. Now, spores, as we showed you in that illustration before, they can be dormant. What does that mean? Dormant is a latent form of the bacterium. They can survive long periods of time in that spore state. They're very, very hard to kill. They usually need these like cocktails of all different sorts to try and destroy them. They're highly resistant to heat, and they're highly resistant to disinfectants, which makes them very, very challenging. Just a few examples of that would be tetanus and botulism. Okay? These both affect the neural system, the neurology. There are small obligate intercellular parasites. These obligate intercellular parasites, it means that they require a living host for replication. Okay, they have this protein coat that's called the capsid, and the protein coat can come in various shapes and sizes, but they can change quickly. When something is changing quickly, we say it mutates. That's important to know. So when it mutates, it can change very, very quickly. It has a nucleic acid core, and that nucleic acid core could either be DNA or RNA. The classification is dependent on the nucleic acid. So it's either a DNA virus or an RNA virus. And some RNA containing viruses contain reverse transcriptase enzyme that can convert RNA to DNA. This is just some different shapes. You can see we used the word capsid before. Here is the DNA or the RNA, and then here's the capsid protective coat around it. Active viral infection, we'll go through some of the steps here, but this illustration of these 6 steps right here are described on this page. So what happens? The virus will attach to the host, right? So here is the virus, and it's attaching to the host and for penetration. The virus enters the host cell, as with active infection. The virus replicates slowly. Oh, let me go back. Sorry, my bad. So the virus attacks to the host cell. The viral genetic material enters the cell, and the viral DNA and RNA take control of the cell. It hijacks it. It uses the host cell to synthesize viral proteins and viral nucleic acids. The new viruses are assembled in the cytoplasm of the cell, and then the virus is released. How is it released? By lysis, by breaking down of the host cell. So we can see that here the virus attaches to the host cell and it penetrates. Then you can see the uncoating taking place here. The viral DNA or the RNA enters the host cell, right? It enters the host cell nucleus, which is here, and it takes control of the host cell DNA. And then the host cell synthesizes the viral components, and it assembles new viruses. And then it releases them, right? You can see the release of many new viruses, and then the host cell lysis, right, breaks down. So that was in the acute viral infection. Latent viral infection, the virus remains in the latent stage. The virus enters the host, as it does with the active infection, but here the virus replicates slowly, or not until much, much later. The viral proteins may also be produced and inserted into the membrane of the host cell, like we saw before. They may stimulate an immune response and destruction of the host cell. Viruses may produce actively if the immune system is depressed or suppressed. We see this a lot, right? Someone may have herpes type 1, but they may not have an active blister on their lip that's oozing, right? We may not see that until what? Until they're under lots of stress, their immune system becomes compromised and weakened, then the virus expresses itself and they end up getting a fever blister, right? When one has a suppressed immune system, it goes from the latent to the active, okay? Mutations, as we said before, some viruses can exist in many forms or different strains. They mutate, they change slightly, right? You usually don't get the same cold twice. It's slightly different. We see this with the cold, we see it with the influenza viruses or the flu. Some viruses can cause cancer. We've seen this happen with HPV, with human papillomavirus. That's why it's cultured. That's why women go for a pap smear, and they look at the different strains. Some are benign strains, and some are cancerous or precancerous. So they have to identify those strains. Let's look at chlamydia, rickettsia, and mycoplasmas. There are some similarities with both bacteria and viruses. They are obligate. Remember that term we spoke about before? Obligate intracellular parasites mean they need a host. These do not grow on an artificial media. They're classified as bacteria, and they replicate by binary fission within the living host. Chlamydia. Now, chlamydia is a very, very common cause of STD, right? Sexually transmitted disease. These typically, as most STDs, are typically reported to the counties or to the states if they come up positive because they're dangerous. They can result in infertility. There are two forms, which we'll go through on the next page. The EB, the elementary body, and the RB, the reticulate body. And when we move to the next slide, you will see that the host cell here, you can see the EB enters the host, and we can see in time that the EB can be transformed into RB. So let's go back here. The elementary body is the infectious form, and it contains a cell wall. So here, this is the infectious body. All right, let's go back. The reticulate body is a non-infectious form located inside the host of the cell, but the reticulate body can convert to elementary bodies, and elementary bodies burst from the host cell, spreading the infection, which we see it goes from the EB, right, the binary fission, the multiplication, converts to the RB. You could see over some time, right? Then we can see it starts to get multiplying, and you can see the RBs, eventually the inclusion, this little encapsulation of the RBs burst, releasing the EBs. And what were the EBs? The elementary bodies are the infectious form, and that's how this STD can spread. Rickettsia is a small gram-negative bacteria, also living inside the host cell, and this is transmitted by insects, right? Insects are known as vectors. That's important to know. Vectors can transmit diseases like lice and ticks. An example of That would be typhoid fever or typhus fever and Rocky Mountain spotted fever. Mycoplasmas, these are the smallest cellular microbe. They do lack a cell wall, and these mycoplasmas can cause some atypical type of pneumonias. Now we'll talk about fungi. Now we spoke about prokaryotic. Fungi are eukaryotic. They contain a nucleus, and they're found throughout the environment. They're found on animals, they're found on plants, humans, food. Fungal infections are sometimes referred to as mycotic infections. They're from single-celled yeast or multicellular molds. Only a few are pathogenic. The cause of the primary infection is found on the skin or mucous membranes, but they can be spread systematically, particularly if the person has, here we see it again, a compromised immune system or they're immunosuppressed. This is seen commonly with people with HIV that get these systemic fungal infections. Here is Candida or yeast overgrowth. This is the mouth. We can commonly see this in the mouth. We see it with people that overuse antibiotics. We see it with people that have high refined carbohydrate diets with all that sugar in their gut, can often cause yeast overgrowth and Candida. We'll see a heavy white coat in the mouth and even on the tongue. There are these anti-Candida diets that are given to try and help people with this, to restore a healthier flora. I'm still moving on with fungi. An example of fungal diseases, histoplasma. These can cause neurological diseases and can be transmitted to the embryo or the fetus if the mother is infected. It can be very dangerous. Tinea pedis, we've often heard, is a fungi of the foot. It's typically called athlete's foot. People can get it in the hands, they can get it in the feet. Sometimes from the locker room you'll hear it, or shared showers in schools and in gyms. Candida, usually harmless, but it is opportunistic and the causative agent of thrush that we saw before, or even vaginitis. Pneumocystis carinii, these are opportunistic organisms that cause pneumonia. Eukaryotic organisms, remember they have a true nucleus. They do lack a cell wall. They're unicellular. They lack a cell wall. Many live independently, and others are obligate parasites. Some examples would be trichomoniasis, commonly sexually transmitted disease. Trichomoniasis is a commonly sexually transmitted disease, or an STD. Another example of a protozoal disease is Plasmodium, which is a causative agent of malaria. And then Entamoeba histolytica, causing amoebic dysentery. Here's a picture of the Trichomonas. Helminths. These are flatworms or roundworms. They're not really considered microorganisms. They are parasites. They could be small, but they could also grow up to about 3 feet long. They have a life cycle that's about 3 stages. They start in an ovum, then it becomes a larva, into an adult. Now they enter the body through the skin or by ingestion, especially if there is fecal matter that's around, that's in the ground. The infections are more commonly found in young kids, and the infection, again, can be life-threatening in individuals who have weakened immune systems or are immunosuppressed. Pinworms, this is if the ova is inhaled in dust because of the fecally contaminated areas. You have to be really careful when sweeping in areas that have, dirty garages, and then or sheds, and you're just sweeping these things, and it creates like this dust cloud. You have to be really, really careful and protect the respiratory tract from breathing in these things. Hookworms. See, if you look at pinworms and you look at hookworms and you look at Ascaris, they're contaminated through fecally contaminated soil or fecally contaminated regions, and that's how they're spread. Have to be very, very careful with those helminths. The Ascaris, these are giant roundworms, and they're ingested with food also that have been grown in feces contaminated soil, or they're prepared by a person whose hands have been in feces contaminated regions. That's why it's really important to wash your hands very well, especially in restaurants, and cooks to wash their hands after using the bathroom. Here's an example of a pinworm on the left. It looks like here is the mouth here, and here is a tapeworm. Now, prions you may not have heard of, but prions are protein-like agents that change the shape of proteins within the host cell. And they are transmitted by contaminated tissues, like the ingestion of meat or infected blood or donor organs. Prions are usually associated with neurodegenerative diseases. It causes degenerative diseases of the neural system. When we get into the neural system, we'll talk about some human prion diseases like Creutzfeldt-Jakob disease or variant of that. So it is a very rapidly progressive disease, and it is not just progressive, but unfortunately fatal. Many areas of the body have resident population of mixed microorganisms. These are called resident flora. They're indigenous. They're the normal flora. It's also referred to as resident microbiota. It's found throughout the body. We have this natural microorganisms on the skin and in the nasal cavity, throughout the mouth, the gut, the vaginal area, the urethra, and they're beneficial. Some produce antimicrobial factors, some produce vitamins, some help even with digestion. Opportunistic infections occur when the resident flora cause an infection. So when the resident flora is transferred to another part of the body, the species of the resident flora become out of balance, right? When you have more bad bacteria than the good bacteria. This happens a lot even when people overuse antibiotics, right? It kills all bacteria, the good and the bad, and when the bad outnumber the good, then we have problems. Especially in the intestines and in the stomach, there is a type of overgrowth called C. diff that becomes very difficult to manage. Opportunistic infections can also be caused with immunodeficiency. Okay, so this may be a good place to pause and go back and review some of those terms, right? Know what pathogens are, and prokaryotes, staph and strep and cocci and bacilli and gram positive, gram stain negative, endotoxins, exotoxins, spores, go over the viral replication, the STDs, helminths. You should, go through some of those basic terms, okay? And now, going on to the principles of infection, What is an infection? Well, a condition where an organism can reproduce in or on the body tissues. And there's a sporadic infection, which is in a single individual. There's an endemic infection, which is continuous transmission within a given population, such as a common cold. And then there's an epidemic infection, which is a higher than normal transmission or spread to new geographical areas, such as the influenza or the flu. And then there's pandemic, right? Transmission has occurred on most continents of the globe. We've seen this with HIV. And then we've learned that term syndemic, which is when you have multiple epidemics happening at the same time, right? We're seeing that with COVID. The transmission of the infection agents, right? You have this reservoir, which is the source of the infection, or a person with an active infection. And that person may be displaying acute symptoms, or the person may be asymptomatic, meaning there's no clinical signs or symptoms. When you have that individual, they're called a carrier. A carrier is a person with subclinical signs of the disease. It could be animals, it could be contaminated water, soil, food, or even equipment, right? These can all spread diseases. So if we look here, we can see you got contaminated water or soil. We can ingest that. Poultry or eggs, or from fomites, right, from these objects. Could be spread from the nasal secretions to the hand, into the lungs, into the mouth, right? Get your hands out of your mouth, so many parents tell their kids, right? It could be, think of how many people share needles, right, with the drug epidemic and heroin epidemic that's happening. It can happen from injury, can happen from insect bites, from animals, STDs, come from so many different sources. The mode of transmission could be direct or indirect. Direct would be touching an infectious lesion or sexual activity when there's contact with infected blood or bodily secretions. That would be direct contact. Indirect contact involves an intermediary object or an organism, right? Contaminated hands or contaminated foods. We mentioned fomites before. That's an inanimate object. Maybe someone's at a restaurant and they cough and sneeze and it ends up on, the silverware. And then you put that in your mouth. Someone coughs or sneezes on their hand and they touch a doorknob, right? So that would be indirect contact. So you should know fomites is also a very important term. We have these droplets, which can be transmission, or even aerosol could be transmission. And they're kind of similar, except, aerosol transmission can be spread a little further because they're lighter. Droplets are a little bit heavier. They could be spread, respiratory or salivary secretions are expelled from an infected individual, like if someone spits or they cough. An aerosol transmission, it involves smaller particles from the respiratory tract, right? There's a difference from speaking to someone or maybe coughing on someone. So the aerosol transmission can be suspended in the air and travel farther than droplets. A vector-borne transmission, right? These are things that can be spread through insects or animals. Those are the intermediate hosts. There's two other terms that we'll go over and we'll show you coming up is the portal of exit and the portal of entry. The portal of exit is the means where the agent leaves the reservoir, which is the source, and the portal of entry is the access in to a new host. Susceptible host or susceptibility depends on a variety of different factors, right? Age, pregnancy, genetic susceptibility, immunodeficiency, poor nutrition, chronic diseases like diabetes, high blood pressure, obesity, and severe physical or emotional stress. Yes, stress is huge. Inflammation or trauma or impaired inflammatory responses. All of these make the individual more susceptible. Okay, let's look at the nosocomial infections. Some call it nosocomial infections. So let's look at the nosocomial infections. These occur in healthcare facilities. That's really important. It occur in healthcare facilities like hospitals, nursing homes, physicians' offices, dental offices. Why? Well, hospitals contain a lot of sick people. Doctors' offices have a lot of sick people. So 10 to 15% of the patients acquire an infection from one of these sources. Okay? In these areas, there's many microbes that are present. In these areas, there are patients that are just undiagnosed with infectious diseases. It's a shared environment. Treatments that may cause weakened immune systems, right? When someone's being treated in the hospital, maybe they're giving immunosuppressants for a flare-up. Maybe they're on chemotherapy for cancer. It weakens them, so they become more prone. And many healthcare workers and fomites act as reservoirs. So nosocomial or nosocomial. Nosocomial or nosocomial. Let's look at the difference between pathogenicity and virulence. So pathogenicity is when there's the capability of a microbe to cause disease. The virulence is the degree of pathogenicity. All right, the degree of pathogenicity. And that has also depending on a few different things. The invasive qualities, right? The motility, are there enzymes present? Are there endotoxins or exotoxins? Is there the adherence to other tissues by pillae or fimbriae or specific receptor sites? And the ability to avoid host defenses, right? Are they hiding from macrophages? So pathogenicity and virulence. Virulence describes the degree of the pathogenicity. Principles of infection. There's always new emerging diseases, antigenetically different forms of common infections, such as the influenza, right? It's always mutating and it's changing year after year. Doesn't seem that they can ever get it right? We always hear, and it's a huge debate whether someone should get the flu shot or shouldn't get the flu shot. There are some people that never ever get the flu shot, never get sick, and then the 1 year they get it, they're in bed for 2 to 3 weeks. I'm involved with the Medical Reserve Corps in my county, and when this topic comes up, there's healthcare providers that swear by it, and there's equally as many that go, I did the shot 1 year and I got so sick, I'll never do it again. I guess like anything else, yeah, I've heard some healthcare providers that say, if you've always done it and you've had no problem, stick with it. And then others say, if you hadn't done it and you haven't gotten sick, stick with it. If you haven't got it, you haven't gotten the flu shot, then don't start getting it. Some say, if you've always gotten it, don't stop getting it. I mean, I've heard so many different degrees of advice with this topic. There are superinfections, which are drug resistant. They're multi-drug resistant forms. These superbugs, they're also called. TB could take 9 months to a year to treat. They use these different cocktails to try and treat it, as well as with Staphylococcus aureus. To try and control the transmission of infection, there are different types of precautions. There are universal precautions. There are guidelines by which all blood, body fluids, and waste products are considered affected. It's just assumed. You know, if someone goes into a hospital and they see an open wound and a lesion, they have to look at that individual, look at all their blood and all their body fluids and any waste as infected. Um, so there are standard precautions, and then there's more specific precautions. So standard precautions, they're used in all settings with all clients when body fluids may be exchanged. People are wearing gloves, they're wearing eye protection, they're wearing masks. Specific precautions, when someone is diagnosed with a particular infection, perhaps they'll say this individual needs to be quarantined for 3 days or 10 days. Okay? And sometimes these are used in conjunction with the standard precautions. To break the chain of infection. Okay, so we have to find the reservoir, right? Locate and remove or isolate the reservoir. What is the reservoir? It's the source. So we'll be taking a look at this, right? Here is the reservoir, the source, the person, the animal, the insect, the environment in the home, the workplace, the community, the bus or the store. Identify and restrict access to contaminated food or water. We've heard many times E. coli infections on contaminated spinach or lettuce or tomatoes, right? So it has to be identified. Reduce contact between the infected person and the remainder of the population. Block the portals of exit and entry. Remove or block the modes of transmission, direct or indirect, and vectors. Reduce host susceptibility by immunizations or adequate nutrition and access to health care. So what we're seeing here is, you can see you have like the reservoir, which is the source, and then it's going to exit, right? There's the portal of exit, and then there's the portal of entry. The portal of exit could be from the mouth, from the skin, from the intestines, and then it gets transmitted, and it could be transmitted directly or indirectly, direct through saliva, blood, feces, semen, or exudate that comes from the skin, or indirect from these droplets on the hands or the surfaces, insect bites, vectors, contaminated food. And with proper hand washing and disinfecting and sterilizing and proper food handling, it can block the entry. Or with barriers, right, from direct barriers like gloves, glasses, gowns, the appropriate and safe removal of waste. Another way of blocking the entry. So everywhere you see these horizontal lines, it's where you can modify or block. So how can you block the entrance into the individual, right? These mechanisms. Otherwise, now it enters the host again, and it can spread from one host into another, right? So to try and prevent that, we talk about immunization, and we talk about proper nutrition and all the proper vitamins and minerals and foods that can help strengthen and optimize one's immune barriers and their immune system. Otherwise, now you have entrance into a host and they're susceptible. And they can be either a carrier, maybe they're asymptomatic and they don't even know about it, so they're just a carrier, or they become symptomatic and they can spread the disease all over again, okay, to another person, animal, insect, home, workplace, and that becomes the reservoir. So this host can be a carrier. They can be in that incubation period, right, because they're asymptomatic, and then they can spread it. So you can break the chain at many areas, okay? Some additional principles of infection in terms of trying to reduce transmission. We have some techniques like sterilization, disinfection, antiseptics. Sterilization is the destruction or removal of all forms of microbial life, including endospores. So sterilization is super important. It's the destruction of the removal of all forms of microbial life, including endospores. There's a technique that are called autoclaves or autoclaves. Um, the equipment has to be cleaned prior to sterilization as well, right? So the sterilizing equipment has to be sterilized. Otherwise, it keeps on spreading and contaminating other objects. Or you can heat it up, right, with a flame. That's incineration to sterilize. Um, Disinfection. That's the destruction of non-endospore pathogens. And disinfection can happen through chemicals. There are chemical disinfectants. There are certain UV lights that can be disinfectants, and boiling. Many parents, when they have infants, they'll put the baby bottle in boiling water to disinfect it. Okay? And antiseptics. The antiseptics are the destruction of non-endospore pathogens on living tissues. So this is when you spray disinfectant or wipe isopropanol or isopropyl alcohol at least 70% on your hands to kill and disinfect. Okay? Good place to pause and review. Pandemic, endemic, what is a reservoir, review fomites and vectors, nosocomial, virulence, pathogenicity, sterilization, all great terms there. In terms of onset and development, the incubation period is the time between entry of an organism into the body and the appearance of the clinical signs of the disease, right? That is the incubation period. This is where things can actually replicate. The prodromal is when they start not to feel too good. They really don't know what it is. It's just, there's fatigue, there's loss of appetite, there's infection. It's really non-specific. They know they're coming down with something, they just don't know, right? The acute period is when the infectious disease develops fully symptomatic, very, very clear. Patterns of infection, there's local infections and focal infections. They're not the same thing, right? Local infections are really confined to one specific area. Focal infections spread from local to another tissue. So I've had a patient a few years back that had a kidney infection, had a bad UTI that started to spread to their ilium, to the lumbar spine, to the pelvic area. Okay, went focal. Systemic infections, there's septicemia, bacteremia, toxemia, and viremia. These all have to do with blood every time you see emia. So septicemia is the multiplication of pathogens in the blood. Bacteremia is the presence of bacteria in the blood. Toxemia, the presence of toxins in the blood. Viremia, the presence of viruses in the blood. Acute versus chronic infections. Some of this should sound familiar. We've done it already. The term acute, sudden onset with severe symptoms, but they're short-lived. The chronic is less severe symptoms, but they last a longer period of time. Primary versus secondary infections. Primary infection is the initial infection, and the secondary follows the primary infection and involves other microbes. Subclinical, when you hear that term subclinical infection, there's no signs or symptoms. So we see here a pathogen enters the host. Here's the number of organisms, and here is over time. So here we have the pathogen. The pathogen, over some time, it can colonize at an appropriate site, and then it really starts to reproduce, and then it multiplies, and then there's the prodromal sign. Maybe they're not feeling too good, and then they become very symptomatic. The acute sign presents. And one of two things happen. It either gets out of control and it continues up to an overwhelming infection, septicemia, and that could lead to death, or they get it under control, decreased reproduction and the death of the pathogen. The host defenses finally kicks in. You have the appropriate nutrients, maybe some glutathione and vitamin C and appropriate zinc. And then they're able to eliminate the waste and cellular debris. Maybe they take antibiotics or antibacterials. And then they start to show some degree of recovery, and the signs start to subside a little bit. And if it doesn't totally resolve, it could become kind of like a more mild chronic infection, but there's no destructive signs. Otherwise, they can go into a total recovery. So signs and symptoms of an infection: if it's the local signs, pain, swelling, redness, warmth. If it's bacterial, remember they have that purulent exudate, and if it's viral, they have a serous, which is a clear exudate. If there are systemic signs of inflammation, the fever can present, fatigue, weakness, headache, nausea. The methods of diagnosis, well, they can culture it and they can stain it. All right. They can use specific clinical specimens, and it also helps with determining what appropriate drug would work best with a particular strain. They can do blood tests. You can get different variations in the number of white blood cells or leukocytes. So if there's a bacterial infection, the white blood cell count goes up. They call it leukocytosis. If there's a viral infection, then leukopenia. Then the white blood cell count goes down. They could do a differential count. They can look for C-reactive protein. They also look at the ESR, the erythrocyte sedimentation rate. All different blood tests to help diagnose the condition. And they can also look to see if their body produced antibodies, right? There's immunological testing of body fluids. They can identify, there's antigen identification, and there's even antibody titers to determine if they have a acute infection or they currently have it, or if they've had a past exposure to it. And that would be the difference between IgMs, meaning they've had a past exposure, or IgGs, they have a current exposure. Drugs should be administered and taken as directed. The antimicrobial drugs should be taken until the prescribed medication is completely used. They shouldn't stop early, right? This happens with a lot of people where maybe they're supposed to take a 10-day antibiotic, and at day 4 or 5 they feel better and they just stop, and then boom, it comes back. You have to be careful. Don't use the prescribed drugs that were given to other clients. You sometimes, or patients, where you're having a certain symptom, you tell a family member or a friend, and they go, "Oh, I have some leftover penicillin. I'll just give it to you." We don't want to do that. Okay. There's also drug resistance known to occur with certain infections. This is when they have to use like a multi-drug cocktail or therapy, typically with tuberculosis and even with HIV. Different classifications of antimicrobials. There's antibiotics. These are powerful drugs that kill or inhibit the growth of microorganisms. A classical example of that would be penicillin, which was derived from mold. And they could be synthetic or even semi-synthetic. Antimicrobials are classified by the type of microbe they treat. They're either antibacterial, they're antiviral, or they're antifungal. Bactericidal versus bacteriostatic. If something is capable of killing bacteria, it's bactericidal. Antibiotics, antiseptics, disinfectants, they're all bactericidal. Bacteriostatic just decreases the rate of reproduction. It slows it down. There are broad spectrum treatment. We've heard of broad spectrum antibiotics or even narrow spectrum. When something is broad spectrum, they're effective in both treating gram-positive and gram-negative strains. That's important with broad spectrum. The narrow spectrum is either specific for either gram-positive or gram-negative. Less likely to affect the normal balance of resistant flora compared to broad spectrum. right? Once you isolate, you know what it is, and you can treat specifically, you do less harm to other parts of the body and other bacteria. First and second-hand generation drugs, pretty straightforward. First generation is the original drug class, and the second generation is just a later version, which may be more effective, it may be more tolerable, it may be more easily administered. They learn from first generation, and they make modifications for second generation. How do these antibacterials work? Really important to know this. They can interfere with the bacterial cell wall synthesis, like penicillin. They can affect the permeability of bacterial cell membrane, polymyxin. It can interfere with protein synthesis, tetracycline. It can interfere with the synthesis of the essential metabolites, sulfonamides. Okay, so I would know these different mechanisms in which these antibacterial drugs work. It can interfere with the cell wall, the bacterial cell wall synthesis. It can increase permeability of the bacterial cell membrane. It can interfere with the protein synthesis, and it can interfere with the synthesis of the essential metabolites. The modes of action of the antivirals drugs may act by blocking the entry into the host cell, inhibiting gene expression, right? DNA and RNA of the virus hijacks the genetics to produce viral components. So it's basically inhibiting the assembly of the virus. Antifungal drugs, they interfere with the mitosis of the fungi. They can increase fungal membrane permeability. And most antifungal agents administered topically, right? Because we'll see them expressed through the skin or mucous membranes. Remember, fungi are eukaryotic cells and are therefore often toxic to animals and human cells. So the treatment requires strict medical supervision. The anti-protozoal drugs, they're similar to the antifungal drugs. Remember, protozoans are also eukaryotic cells. They require treatment with different agents at different stages of the cycle. Okay, so we hope you enjoyed that lecture.